Hi everyone. Today's lecture is on part 1 of amino acid metabolism and urea cycle. Learning objectives. List the metabolic functions of protein and amino acids. Explain the importance of proteins in the human diet and define incomplete protein, complete protein, essential amino acid and non-essential amino acids. Define the amino acid pool. Define nitrogen balance and state under what conditions a positive and a negative nitrogen balance occurs. Define each of the following reactions in protein metabolism and state their importance: transamination and oxidative deamination. Explain the importance of urea cycle and its regulation. State the importance of the interconversion of amino acids and intermediates of carbohydrate and lipid metabolism. Distinguish between ketogenic and glucogenic amino acids. Metabolic functions of proteins and amino acids. Protein function as enzyme, many hormones and antibodies. Hemoglobin and cytochromes are also proteins. Some proteins involve in supportive function and many other involve in movement. Amino acids form hormones and neurotransmitters. Example: thyroxine, epinephrine, dopamine. Amino acid can be a source of energy. Dietary protein contains about the same amount of energy per gram as carbohydrates. The non-nitrogen part of amino acids can be oxidized directly in cell respiration or after conversion to ketone bodies. Excessive use of amino acid for energy causes a deficiency of tissue proteins and an excess of ketone bodies. Dietary protein is the source of essential amino acids. Dietary proteins provide the amino acid that human cannot synthesize. These are known as essential amino acids. The non-essential amino acid can be synthesized endogenously from intermediates of glycolysis or the TCA cycle. The essential amino acids are arginine for children only, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. All other amino acids comes under non-essential amino acids. The mnemonic used to remember essential amino acid is PVT Tim Hall. In biochemical terms, non-essential amino acids have synthetic pathways in the body. For essential amino acid, there is no synthetic pathway present in the body. In nutritional terms, dispensable amino acid. These amino acids can be excluded from the diet without affecting nitrogen balance. Indispensable amino acids, when they excluded from the diet, results in negative nitrogen balance. Example: isoleucine, leucine, lysine, threonine, methionine, valine, phenylalanine, and tryptophan. Conditionally essential amino acid. Some amino acids become essential only under certain conditions. Otherwise, they are classified as non-essentials. Rate of synthesis of glutamine, cysteine, and arginine not sufficient after trauma, major surgery, and during sepsis. These are known as conditionally essential amino acids. Tyrosine and cysteine are synthesized from essential amino acids, phenylalanine and methionine respectively. Phenylalanine and methionine are indispensable amino acids. If phenylalanine and methionine present in the diet or below minimal requirement, then tyrosine and cysteine become indispensable, in other words, conditionally essential. Amino acid can be categorized as glucogenic, glucogenic and ketogenic only. The glucogenic amino acids, which are non-essential, is given in the table. Methionine, threonine, valine are the essential amino acids which are glucogenic. Tyrosine is a non-essential amino acid. It can be glucogenic and ketogenic. The essential amino acids isoleucine, phenylalanine, and tryptophan are glucogenic and ketogenic. Leucine and lysine, these two amino acids are essential amino acids. They are only ketogenic. Protein digestion in the GI tract. Protein digestion begins in the stomach when the acidic environment favors the denaturation of proteins into random coils. Denatured proteins are more accessible as substrates for proteolysis than are native proteins. The primary proteolytic enzyme of the stomach is pepsin, a non-specific protease that remarkably is maximally active at pH 2. Thus, pepsin can function in the highly acidic environment of the stomach that disables other proteins. Protein degradation continues in the lumen of the intestine. The pancreas secretes a variety of proteolytic enzymes into the intestinal lumen as inactive zymogens that are then converted into active enzymes. The battery of enzyme displays a wide array of specificity, and so the substrates are degraded into free amino acid as well as di and tripeptides. Digestion is further enhanced by proteolytic enzymes such as aminopeptidase that are located in the plasma membrane of the intestinal cells. Aminopeptidases digest proteins from the amino terminal end. Single amino acids as well as di and tripeptides are transported into the intestinal cells from the lumen and subsequently released into the blood for absorption by other tissues. Protein absorption as amino acids. Protein digestion primarily a result of the activity of enzyme secreted by the pancreas. Aminopeptidases associated with the intestinal epithelium further digest proteins. The amino acid and di and tripeptides are absorbed into the intestinal cells by specific transporters. Free amino acids are then released into the blood for use by other tissues. Transepithelial amino acid transport. Amino acids are absorbed from the intestinal lumen by semi-specific sodium-dependent transporters, some with overlapping specificities. The same transporters are found in the renal epithelium and facilitate reabsorption of amino acids. Enterocytes can also absorb and proteolyze small peptides. On the serosal side, amino acid transport is by facilitated diffusion. The low intracellular sodium ions is generated by sodium-potassium ATPase. Protein turnover. Protein turnover, the degradation and resynthesis of proteins, take place constantly in cells. Although some proteins are very stable, many proteins are short-lived, particularly those that participate in metabolic regulation. The proteins can be quickly degraded to activate or shut down a signaling pathway. In addition, cell must eliminate damaged proteins. Why should protein turnover occur at all? Abnormal proteins degraded and replaced by newly synthesized proteins. Abnormal proteins arise in cells due to spontaneous denaturation, errors in protein synthesis, errors in post-translational processing, failure of the correct folding of the protein, damage by the free radicals. Turnover helps to maintain concentrations of free amino acids both within the cell and in the blood. Maintenance of free amino acid pool. The blood amino acids are derived from two sources, dietary protein and endogenous protein. These amino acids are utilized to synthesize new proteins in the body. Purines, pyrimidines, heme, neurotransmitters, hormones, and other functional nitrogen products also synthesized from these blood amino acids. The nitrogen part of amino acids, or the amino nitrogen, can be converted to urea and eliminated or excreted in the urine. The carbon skeleton of the amino acid either enter the glycolytic pathway or TCA cycle to produce ATP and carbon dioxide, or it can be utilized to synthesize glucose and subsequently glycogen. Carbon skeleton also can be converted to lipids in lipid biosynthesis. Nitrogen balance. Nitrogen balance is the difference between the nitrogen entering the body and leaving the body. Nitrogen equilibrium means the nitrogen intake equals nitrogen output in urine in the form of ammonia, urea, and uric acid. Nitrogen balance is controlled by diet and hormones. Normally, hormones control protein catabolism and anabolism to maintain nitrogen balance. If the nitrogen intake is equal to nitrogen loss, it is said to be nitrogen equilibrium or nitrogen balance. If the nitrogen intake is greater than nitrogen loss, it is referred as positive nitrogen balance. If the nitrogen loss is greater than nitrogen intake, it is said to be negative nitrogen balance. The normal healthy adult, the nitrogen intake is equal to nitrogen loss. That means a normal healthy adult is in nitrogen equilibrium. Some of the conditions like growth, pregnancy, bodybuilding, patients recovering from severe illness are example for positive nitrogen balance, where nitrogen intake is greater than nitrogen loss. Negative nitrogen balance observed in protein deficiency and essential amino acid deficiency. Wasting diseases, burns, trauma, and chronic diseases are examples of negative nitrogen balance. Half-life of proteins. Regulatory proteins, for example, digestive enzyme, lactate dehydrogenase, or RNA polymerase have short half-lives, minutes to hours. Structural proteins, for example, collagen, have long half-lives and last for years. Hemoglobin has an intermediate half-life of about 120 days. Half-life of proteins correlate with their amino terminal residues. Proteins with N-terminal residues, methionine, serine, threonine, valine, or glycine have half-lives greater than 20 hours, and proteins with N-terminal residues phenylalanine, aspartate, lysine, leucine, or arginine have half-lives of 3 minutes or less. Proteins rich in proline, glutamate, serine, and threonine are more rapidly degraded than other proteins. Protein degradation can take place by three mechanisms. Proteolysis of dietary proteins in the stomach and lumen of the small intestine releases free amino acid into the bloodstream. Proteolysis of proteins that move through the endocytic pathway takes place in the lysosome of all cells. Controlled proteolysis of ubiquitin tract intracellular proteins take place in the proteasome of all cells. Protein degradation. To avoid immune response, foreign dietary proteins are degraded outside the body within the lumen of the gastrointestinal tract. Two groups of cell protein, extracellular, intracellular, are degraded via distinct mechanisms. Extracellular proteins are blood coagulation factors, immunoglobulins, albumins, cargo-carrying proteins, and peptide hormones. Extracellular proteins degraded by lysosomal degradation. Intracellular proteins are degraded by ubiquitin-mediated degradation. Protein degradation, three possible pathways. Ubiquitin proteasome system, lysosomal autophagic system, calpain-calpastatin system. In the lysosomal autophagic system, the extracellular proteins internalize by endocytosis and form endosome inside the cell. These endosome fuses with lysosome and form an intermediate compartment known as endolysosome. The lysosome proteases hydrolyze and degrade the proteins. Lysosomal autophagic system. Lysosome contain approximately 40 enzymes capable of hydrolyzing all major cellular components. A variety of proteases, cathepsins, and peptidases to hydrolyze proteins. The pH within the lysosome is 4.5 to 5, and all lysosomal enzymes exhibit low pH optima. This ensures if they leak into the cytosol, their activity is low and little damage is done. The low pH within the lysosome is maintained by a proton pump driven by the hydrolysis of ATP, thus contributing energy requirements of degradation. Ubiquitin proteasome system, located in the cytosol and nucleus, degrades abnormal proteins and short-lived cytosolic proteins. It is an ATP-dependent process. Proteins are targeted for degradation and tagged by conjugation within ubiquitin. Proteins targeted by ubiquitin: cell cycle regulators, tumor suppressors, growth regulators, cell surface receptors, mutant and damaged proteins, transcriptional activators or inhibitors. Ubiquitination, the molecular kiss of death. In general, Multiple ubiquitin units are arranged in polyubiquitin chains linked via lysine 48 of ubiquitin targeting the protein for degradation. The target protein identified and then ubiquitin attached to the target protein at the expense of ATP hydrolysis. Subsequently, further ubiquitin molecules attached to the existing ubiquitin molecule and the protein is stacked and referred as ubiquitinated protein. The ubiquitinated protein is presented to the proteasome, which is having proteolytic activity, and hydrolyze the protein into peptides and amino acids. The released ubiquitin is utilized in another cycle of protein degradation. Calpain calpastatin system. Calpain are enzymes that consist of proteolytic subunit and a calcium binding subunit. In the cytosol, these enzymes are inactive due to binding of inhibitory protein calpastatin. Attachment to the cell membrane remove this inhibition and activation occurs at low concentration of calcium ions. The enzyme hydrolyzes proteins to peptides. Complete hydrolysis requires peptidases. Regulation of protein degradation. Ubiquitin proteasome system controlled by concentration of ubiquitin and activity of proteasome complex. Lysosomal degradation controlled by number of particles transported into cell. The calpain calpastatin system controlled by intracellular calcium ion concentrations. Defects in protein degradation and diseases. Failure to control the rate of degradation of cyclins could lead to their overexpression, increasing the risk of tumor development. Muscular dystrophies are characterized by variable degree of muscle weakness and degradation. Example, Duchenne muscular dystrophy and Becker muscular dystrophy. Neurodegenerative diseases such as Alzheimer's, Parkinson's, motor neuron disease, and spongiform encephalopathies is the accumulation of insoluble protein which form aggregates in neurons. Thank you.